you. Well, welcome everyone. I changed the title just slightly because it's not just about genetics that I'm going to be talking about. It's sociogenomics. So that's why I was so excited about the title of this conference, because what I do is I look at the genetics of sexual and reproductive behavior, but across time and space, which is the socioeconomic element. So I apologize for some of you that have sort of seen me introduce genetics, but there are some other people that may not know what this whole field is, so I'm going to give you a brief introduction as well. So, and with gracious for the funding from the European Research Council that will soon not be possible for people from the UK, I'm assuming. So congratulations to everyone from Europe that will be able to take that money. Okay. So I don't think, this is the nice part about a fertility conference, because I've already seen a lot of these graphs, so I don't even need to give the introduction about what's been happening with fertility that I normally do. But this is a topic that's dear to my heart that I've been studying for quite some time. So we know that, we've had drops, and this is completed cohort fertility, and I often use the Human Fertility Database in all my research. But I want you to take a look at Iceland and the US. We're going to come back to that in a minute, because these are two pivotal countries for genetics, and I'll tell you about that in a moment. And another thing we've seen, and this is work from Thomas, that we've had this increase in childlessness. It's a U-shaped curve, but it's also increased in certain regions in the world. So these are things that we've been noticing as demographers. And another thing we know as demographers is that we've had this really rapid increase in the mean age at first birth across many countries. It's been as much as 4 to 6 years for the mean age at first birth. So this is mean age at first birth for women for various countries across time. So if you think about it, women and couples are now having children around age 29 or 30, and this is exactly the period where many individuals start having problems with conceiving. So we're having, longer times to conception, and they're starting to have fertility problems. So for this reason, I started to think, wait a minute, shouldn't we look at more the biological and the genetic factors that might be at play? So that's sort of what started this research idea. And I did some reviews with Ron and Peter, but also with Francesco and a former PhD student of mine, Nicoletta. And I think I don't even need to tell this crowd, about what the main drivers are for postponement of having children and lower numbers of children. Things such as contraception, which Francesco talked about yesterday, changing values, educational expansion of women. We're going to have talks about education here, just right after my talk. Work-life balance, the motherhood wage penalty, gender equality. These are a lot of the things we knew and we know about. And I was sort of studying this topic, and I was talking to some biology friends of mine, and we were out having some drinks, and they were making some jokes about me, saying, oh, this is Melinda, she studies fertility, but, they're very socially deterministic. They don't think it has any biological component, and everybody would just laugh. They thought it was really, really funny that demographers often study fertility. Although we study mortality with a biological component, we often don't look at the genetic or the biological aspects. So we know that fertility, diseases, social behavior, it's influenced. So these are the things that normally that we've studied. In psychology, they've looked a lot more at choice and individual and person characteristics. But within genetics and reproductive medicine, there's been a lot more focus on the reproductive span, and we saw that yesterday in Michelle's paper from Sardinia, these reproductive periods, and sperm production and ovulation. So the idea of this project that I'm in the last year of right now, but it'll probably go on for quite a long time after that, is to move from, this is what I've done for most of my career, and now I've focused on this, and I'm going to present you some of these results. So to not just look at the direct effects of social factors, or the direct effects of genetic factors, but to actually see if we can get some measures of the genetic factors leading to age at first birth, sexual behavior, a number of children ever born, use those in statistical models and look at the interaction. So if you have a particular genetic predisposition to having a late or early first child, and or a number of children, or a genetic predisposition to have infertility problems at a certain age. Does that matter across time and space in different social environments? So I'm going to go through this quickly because I want to leave some time for discussion, and we started a bit late, but I had to really laugh when I first started. So one thing I noticed as we were just looking at fertility, and I thought, oh, it would be really interesting, because I have a hunch, and we have a hunch in our research group that sex might be related to fertility. So we thought, okay, let's start looking at this. And if I was an alien, and I came in and I thought, okay, let's look at demographic literature to see if I could get sort of an understanding of what demographers know about sex and sexual behavior, I wouldn't learn very much. So I'm going to go through this quickly, but I just found it so funny that I might even write it up. So I looked at sex and fertility, and what do you think? What do we understand about sex? Well, it's sex ratio. That's our big one. We divide things by age and sex. We look at sex selection and honorable mention for same-sex couples. So, you know, these are the primary things if you look into demography. And then I thought, okay, I've got to get more explicit here, you know, so then I searched for sexual intercourse. And, you know, it's a lot about prevention, so we're really interested in stopping it in any way possible. Pregnancies and HIV, you know, and lots of sort of negative things, father absence, violent conflict. Even migration, but you know, it's really funny how there's not that much of a connection between sexual behavior and fertility and demography. So that's an area that I'm currently expanding in now. I had sort of a disturbing discovery that Germaine Greer, at least I find it a provocative book cover. The only book I could find that had sex and fertility in the title was this now well-known book by Germaine Greer. But otherwise, it's quite negative, sex and fertility on crack cocaine, gallstones. So it's really interesting how we haven't really looked that much at sexual behavior. If we look across different countries, we see that there's actually quite a difference in the age at first sexual intercourse. So from Spain being one of the highest to, again, Iceland being one of the lowest. I have a 16-year-old daughter, so this is quite a stressful topic for me as well to study. So you see this, and we've seen some changes across time, but it's really something that we haven't looked at very much. So what I decided to do was to start looking at the genetics of sexual behavior and the reproductive window and period. So we look at age at first sexual intercourse, age at first birth, number of children ever born, and childlessness. But I also link it very much with the reproductive window. So looking at age at menarche, voice breaking in boys, I realize it's not a great measure, but it's what we have for men. So men are always a problem in fertility, as you know. And age at menopause. So you might know about some of the traditional ways that we've looked at genetics and fertility. And one is behavior genetics, looking at similarities between monozygotic twins. So they have about 100%, almost 100% similar genetic material. So they would compare that with dizygotic twins that would have about a similarity of siblings, you know, their genetic similarity. And they would try to piece out, okay, what's related to genetics, what's related to the shared family environment, and what's related to everything else. So this was, you know, and if you hear these statistics about, you know, something's 10% heritable or 20% heritable, it's usually from these sorts of studies. So that's, although I have done that, that's not what I'm doing. I'm doing molecular genetics. And what that is looking actually at genetic loci. They're called SNPs, or single nucleotide polymorphisms. And what we do is we go beyond just saying, okay, something's a certain 10% heritable, to actually isolating the genes. What are these genes? What are these genetic loci? And what is their biological function? So what do they do? And we know from Hans-Peter Kohler and Miller and Rogers and all of these people that started to look at this at the end of the 90s that this shows you the heritability, just a summary from twin studies. You see it across different countries, you see it across different cohorts, time and space, again, always comes back. And this is for age at first birth, the number of children ever born for men and women. Since it's sort of, this is from 0 to about 0.5, so 50%. But just to summarize, so what you see is there's, the twin studies suggest that there should be a genetic basis to age at first birth and number of children ever born. In fact, they're suggesting from the similarities of twins, it should be anywhere from 25 to 50%. But what you also see from this, there's some wildly different levels of heritability across cohorts and across country. And we virtually have no studies of men, so this is one thing that I wanted to include as well. And I don't even need to talk to people in this room about the issues we have with collecting data from men and how that's been difficult. If you're interested, I'm just going to go through this quickly about how these methods have resolved. We have quite an accessible commentary in the Proceedings of the National Academy of Sciences. Talked about how we've moved from looking at these different methods to the genetic profile scores we use now. So we first started to look at it. I did it together also with Felix, and we moved, so when you move from looking at twins, there's a new method called GREMO. That's genetic restricted estimated maximum likelihood methods. And what you can do is you can actually, if you have genetic data of a population, you can look and see how related you are with anyone in the data set. So Thomas and I might be 40% genetically related, maybe. And Ava and I might be 60% genetically, that would be high. But we have the same color hair, so we might be also, so there's a variation in people across data sets to see how related you are. So we used that new technique that was developed in 2010. And from that, we were able to say, looking at unrelated individuals across various countries in these large data sets, that there was about a 15% basis using whole genome, actual genetic data, to predict, so age at first birth, and 10% for number of children ever born. So this suggests that we've been missing one of the explanations that are related to age at first birth and number of children ever born in our demographic research. But then you get to, okay, yeah, it's 10, 15, 10%, so who cares? So what? You still are at this, it's 15 or 10%, but you don't know what it is, you don't know what the genes are, and you don't know what their biological function is. So in that case, we engaged in a Genomewide Association study. And what that is that you look at genetic data across which is millions and millions of regressions. It's actually just dummy regressions across chromosomes. At first I was amazed by it. I thought, oh, this is so exciting, but it's really just ordinary least squares regression. But, what you do is it's data mining across all these single nucleotide polymorphisms, or what the geneticists call is hypothesis-free research or agnostic research, but we call it in social sciences data mining. And what you do is you have two different replication centers, and I think this is really interesting. So we do our analysis in one place, but somebody does the exact same analysis in another place, and you come together to make sure you get the same results. That's required now within this field. It'd be great if we could start doing that, because it's actually quite fun to come together and discuss it. So we published that in Nature Genetics in 2016, and Nicola was also working on the project with me as well. And we identified 12 loci. I have to just have a little gripe here because, we've been trying to bring this research into demography, and I've submitted this and our new study to the PAA, Population Association of America, and I've never had it accepted for presentation. They found it too weird. But luckily, Nature Genetics, impact factor 39, liked it. Anyway, so that was just my gripe. It was a fairly large sample size, so what you have to do is you combine like over 60 data sets together, and I got them from everywhere. I got them from 23andMe, insurance companies, scientists, and that's the only way you can get a large sample. And as you can see, we have a really the largest male sample to date on this topic. We did discover different loci, and I'll just go through this quickly. These are what's called Manhattan plots. This is the chromosomes, and this shows the significance level. Due to multiple testing, we have very stringent criteria. So you can see these spikes are the genetic loci that we found. We identified, then we returned the results over to molecular biologists, and we found some really interesting things, particularly in relation to male infertility. So we know all the ART and the IVF is focused on women, but this suggests that people that are having particularly 0 children or very late children, particularly with men, there's issues with sperm mobility and motility, also follicle stimulating hormone. I won't go through all of them, but we found some really interesting downstream biological work. So what's being followed up by experts in this area is that we seem to isolate genes that related to lower sperm quality and count. And this is interesting because the medical studies looking at some of the medical aspects didn't find this, but when we were looking at behavioral aspects, we actually uncovered it. Another interesting thing we found is it was very much linked to human development, and it was linked to these, so this is why when Michelle Poulin was describing these reproductive periods yesterday, I was getting quite excited. You went through it too fast for me, but you know, we have this shifting of reproductive periods. It'd be really interesting to think about that more. This is LD score regression that compares other genetic-wide association results. So this is the overlap in genes, basically. So there's a high overlap with age at first birth, with age at menarche and menopause. So there seems to be a lot of shared genetic and biological foundation to this. If you have a later age at first birth, it's linked to later timing of puberty as well. So it's really interesting to think of this in a developmental way. What we're discovering in our next study that we're finishing now is this very much related to risk too. So there's a large overpinning not only with development, but there's a GWAS coming out in Nature Genetics on risk behavior, and there's also one coming out on certain personality self-control. It's very much related to this as well too. So what we were finding is that with certain individuals, they seem to have a shift from later age at menarche, first sex, birth, and then what's really exciting in a study that we're predicting out now is that we're using these polygenic scores to look at their mother and father's age at death, but also using death records. And these scores seem to predict out sort of a longer life period, and it's related to mortality and life expectancy, which is really interesting. We have a new study now where we've expanded it to look at childlessness and age at first sex. We're using much denser data, and maybe another thing I should say is you always, before you do these analyses, you have to post your analysis plan on Open Science Framework. It'd be interesting if we had to do that too. So it means you can't fish for results. You have to do the analysis plan that you said. I guess the only way I could describe what we're doing now is almost like a pixelation. So this was the study, the really old study we did in 2016. And this is the new study where you just get a lot tighter confidence intervals and just a much better measurement because of the newly imputed data. So working in this field is like sitting on a bullet train. You know, it's just, it moves really quickly. New techniques come out almost monthly. So you have to really be agile. So in the new one that we're finishing now, we have huge samples. I just realized I can look up there too. So it's about 700,000 for number of children ever born. And we've got really large sample sizes for men, and we've expanded it out. We found a lot of new loci, and I'm really excited about it. This is still under embargo, but for age at first sex, we have 271 loci, so this is a really strong genetic score. Also for age at first birth, number of children ever born, and we also looked at childlessness as well, just to find out if we could piece out something that would be unique, even though it's very related to number of children ever born. I won't go through this in detail, but we've just got many more stronger hits. This is age at first birth. So what do we do with these results? Okay, so then you have this genetic-wide search. You have these 271 genetic loci, and there's other ones that are related as well. So what you do is you create a variable, a polygenic risk score. And the only way that would be similar, I think, for you to think about, let's say you have a question about attitudes about housework, and you combine those 5 questions into one variable. It's sort of like that, but so it's one quantitative trait that you can use and you can add into your statistical models. So those are called polygenic scores, and they're weighted averages. So what do we have now? So this is the results that will be coming out. Well, we're actually able to predict quite a bit. In some samples, up to 6% for age at first sex, and around 3 and 1.5% for number of children ever born. And I know you're all sitting back and saying, that doesn't sound like very big effects. But I would then just ask you, do you know, if you entered one of your variables in your last model, education for example, do you know what the effect size of one direct effect was? Do you know what your R squared was in your last model? You'll be shocked when you go back and look at how much variance you explained, because we really don't explain that much. So just to put it into perspective, if you look, if I just entered direct effects, usual suspects, we have, the age at your partnership, educational attainment, labor force participation. You can see that our polygenic scores are actually getting fairly close. They'll always remain lower than social variables, and I can talk about that if we have some time for questions too. So we wouldn't expect that these genetic factors would be more important than social behavioral factors, because these are behavioral outcomes. But they're still quite important, and it shows we've been missing something in our research. So, and also when we add them into multivariate models, you can see, and I'll just go through this quickly, but they explain maybe around, this is the Wisconsin longitudinal study, and we're looking at childlessness. But, these variables, and I've added in different genetic variables, they don't predict as much as the social science indicators, but they're still viable in different models. So now I just want to move to conclude quickly talking about how I think these genes differ across time and space. So this is my mom. And this is the social control in the room. So this is her mother-in-law. This just shows when she had her first child. This is actually my second child, so I'm cheating a bit. But we had our children in just completely different environments. There was a lot of social control in her environment, a lot of what Francesco talked about yesterday, the social timing of children and when you should have a child, whereas I was in a period where I had relatively little control, I had lots of control over when I could time and have my child. So it started to bug me, looking at these different genetic studies, that we were combining 63 different data sets across 20 different countries, all of these different cohorts, everything that, that a demographer should not do. And I knew that this was happening, so wouldn't different genes be important across different time periods? And wouldn't they be important? So she was having her children in rural Canada, and I was having mine in the Netherlands, so wouldn't these contexts make a difference? So we published in Nature Human Behavior, using the Human Fertility Database, looking at this across different countries and across time. And I think I'm going to just go through it quickly, because I can see where we're getting for time. But if you're interested in that, you can look at, and this got a lot of resistance from the genetics community, as you can imagine, because we showed in this study that actually, there was an issue when you combined all of these countries together and all of these different cohorts together. And we showed via various simulations and actually real data that perhaps for height and biological traits it wasn't a problem, but it was definitely a problem, and we were missing a lot of the heritability if we looked at social traits. I can talk about that later if you like. In a new study that we're about to publish too, we're showing the genetic heritability, not using twin data, but actual whole genome data, that it differs. These are different cohorts. This is the mean age at first birth from the Human Fertility Database. And also looking at the actual genetic heritability. So it indeed seems to be more realized in the younger cohorts. With Renska, we're also looking at age. And this is another factor that's quite important. So we're realizing that some of these factors related to endometriosis, polycystic ovarian syndrome, but also the ones related to childlessness only seem to kick in, their importance kicks in at about age 35 or later. So this is really important. And then other factors such as education seem to be protective against these perhaps negative genes. And And just one of the final things, this is a work I'm doing with a computational biologist, David Brazell, and he's in our group now as well. And we wanted to look at the genetic overlap between all of these different traits. And indeed, we have a very strong genetic overlap with age at first sex and age at first birth. But something else we're figuring out, too, is the genes that predict, and this is shocking for some, but people know about this in the genetics world, the genes that predict educational attainment and cognitive ability, there's also a strong overlap with age at first birth. The correlation is actually 0.73 between the two genetic scores. So this is something we're doing genomic structural equation models to piece out what's related to education, what's related to fertility and infertility. So to conclude, fertility is in the genes. So Hans-Peter Kohler asked in 1999, is it? And yes, it is. And we actually know which ones now. So I would just like to say that we offer these new variables. They're about to be added in to about 15 different data sets that have genetic data. So it's quite standard in some of the American data sets to have these data, and it's increasingly in the UK as well too. We show that there's a relationship between sex and fertility that we're going to add in, particularly genetically as well. And one thing I don't have time to talk about, but we're working with David Reich's group, and we're actually now tracing these genetic paths from ancient genome data as well too, to see how there's been some selection over time. And now I want to just conclude with something really critical, because there's a sociologist in me who will pretty much break down everything I've just discussed. I would like to talk about some critical thoughts. So you enter as a social scientist into this genetic world. And we've done an analysis of the 4,000 Genomewide Association studies to date. So when you see a gene discovered for this or that, there's a whole bunch of discussion about it yesterday in the media as well. And there's a lot of excitement, and this is where a lot of the science funding goes to. So, but from entering this field as a social scientist, you realize it's quite a unique world. So we looked at it, and we've got a study that's forthcoming any day now in communications biology, where we actually do a computational analysis of all of the genetic wide association searches to date. The first, you might have heard about this before, but the first disturbing aspect about this is throughout time, this shows the genetic wide associations throughout time. The bubble represents the sample size, so we're up to 1 million. And this represents different ancestry groups. Blue represents European ancestry, and red represents Asian populations. But what you should see here is this is very, very white research. We also looked at all of the publications, and we looked at all of the data sets that were used. And not only is it largely European ancestry, they're healthy, they're generally older, overrepresented by women. So this will be coming out soon. Also rejected, of course, 5 times by genetic journals. But we'll get it out. It doesn't have a very nice message, I think. Another thing that's shocking, remember I showed you Iceland and the US? 72% of all genetic discoveries to date come from respondents in 3 countries. I just want to let that fall, because that was shocking when we found that out too. So this is really interesting when we're thinking about variation across time and space. And drugs are being developed. This is where all the pharmaceutical companies' drugs are being developed from these things. And we know as demographers that these things differ across time and space. So that's just a few of the publications. And if you're interested in our research, you can go to this website. So I hope I was short enough, Thomas. So thank you very much.